We're going to discuss your recent study looking at CD14 positive monocytes in rheumatoid arthritis. But could I start by asking you to introduce yourselves?
I'm Professor Ursula Fearon, and I'm Head of Molecular Rheumatology in the Trinity Biosciences Institute in Trinity College, Dublin. And basically our group is focused on investigating the pathogenic mechanisms involved in driving autoimmune diseases, specifically rheumatoid arthritis and psoriatic arthritis.
Hello, my name is Hanlon, Dr. Hanlon. I am a postdoctoral research fellow in Professor Ursula Fearon's Molecular Rheumatology Research Group here in Trinity College, Dublin. And so my PhD research and continuing on into my postdoctoral studies has been focused on the myeloid compartment of the synovial joint. So although there are a lot of cell types within the synovial joint, my research focus has been on myeloid cells, including monocytes and macrophages.
And I'd just like to emphasize there that our research is very patient focused. So what we call is translational research, bench to bedside approach. And just to emphasize that there's three partners in our research. There's the clinical team, there's the scientific team, but most importantly, there's the patient, because without the patient, we actually couldn't do this type of research. So I really want to highlight their involvement and engagement in research.
Could you tell us a bit about the background to this study and what was already known about rheumatoid arthritis?
About 20 years ago, on the right-hand side there, number 3, where we have chronic rheumatoid arthritis, that's where we treated patients 20 years ago, where they had really established ORA and functional disability. So it was way too late and patients' joints were destroyed. Then with great medication over the last 20 years, we started to treat patients much earlier here at the clinical onset. And then more recently, which is kind of the focus of some of this paper, is we're starting to look at individuals, actually, not patients, individuals pre-disease and trying to predict whether they will develop rheumatoid arthritis. So in this paper, we looked at two cohorts, basically. We looked at patients with established ORA, and then we looked at a second cohort, and these are individuals at risk of developing ORA. So just to maybe give you a bit of background on both cohorts, basically rheumatoid arthritis is a chronic autoimmune disease characterized by systemic inflammation. And at the same time, we get new blood vessels in the synovium, which is the tissue in the joint. And these new blood vessels allow these immune cells to migrate into the joint. They activate inflammation and cause cartilage and bone destruction and functional disability. Now, we don't know what the trigger for rheumatoid arthritis is, but we know it's a combination of genetic and environmental factors. And it's this combination that causes a change in a protein on our own cells and our own tissue. And that, causes our immune system to mistakenly attack ourselves. Normally our immune system attacks foreign pathogens that come in like viruses and bacteria, whereas in autoimmune diseases, our immune system attacks ourselves. So what it causes is our immune cells in our circulation to become activated and proliferate. And immune cells like the monocyte, the T cell and the B cell, and the B cell produces autoantibodies, which is very important in the context of this study. And at the bottom of the screen there, you can see that one of those autoantibodies is ACPA, which is present in about 70% of patients with rheumatoid arthritis. And this helps us diagnose rheumatoid arthritis from other forms of inflammatory arthritis. So once these cells in our circulation are activated, they get a second signal that instructs them to migrate to the joint, where they basically set up shop in the synovial membrane. They produce potent inflammatory mediators that cause the inflammation. And so I suppose the context of those autoantibodies brings me to the second cohort. And these are patients that don't have rheumatoid arthritis at all. They could be any one of the three of us here on this call. So basically, these are individuals that present to their GP. They have aches and pains. They don't have any clinical manifestations of rheumatoid arthritis, but what they do have is these autoantibodies in their blood. So we reckon that these individuals have the genetic background. They've had some environmental insult, like they might smoke, or they might have had some pathogen that caused the insult, but they didn't get that second signal to cause those cells to migrate to the joint to develop rheumatoid arthritis. So that's kind of the focus of this study, to try and figure out, can we find a signature in the patients with rheumatoid arthritis that possibly is also present in these individuals pre-disease, and can we predict when they might develop rheumatoid arthritis? So that's our 2 cohorts, and that was kind of the premise of the study.
Could you tell us a bit more about monocytes in this context?
I mean, if you're in my lab, depending on who you spoke to, they'd have a favorite cell. And Megan's and mine at the moment is the monocyte. But the monocyte is really, really important because it's one of the first critical innate effector cells central to driving rheumatoid arthritis. And I'm just going to really point out 3 main functions. Its first function is that they phagocytose antigens, so they try and get rid of, bad pathogens that come in. The second one is that they produce lots of pro-inflammatory mediators. And the third, which is really important in this context, that when they get that second signal, when they're in the blood, to, toddle off to the joints and set up shop there, once they get environmental influences in the joints, they can actually change into 3 different cell types. They can change into a dendritic cell, a macrophage, and an osteoblast. And those 3 cells are really important in further driving the inflammation in the joint. And the dendritic cell and the macrophage, cause the synovial inflammation to invade the cartilage, but that third cell, the osteoblast, causes basically holes to be eaten in your bone, and this causes the dysfunction. So they're really central to both the systemic and the actual joint inflammation to driving this disease. So they're really important. So that's why we focused on the monocytes.
We're discussing your recent study looking at CD14 positive monocytes in rheumatoid arthritis. Describe for us what this study was designed to investigate.
So basically, we isolated this monocyte from the blood of patients with rheumatoid arthritis, from individuals at risk of developing rheumatoid arthritis, but we also compared this to healthy controls just to check that they were still higher than the healthy controls. And there was 3 aims. The first aim was to utilize these monocytes to see, could we identify a signature of cellular dysfunction in established ORA. The second was to see, well, if this identified signature is a signature of established ORA, is it already present also in these individuals at risk? And the third was that there's this really huge body of evidence and, really exciting field in immunology of metabolism. And we now know that immune cells, when they become activated in these diseases, they change the way they use energy. So once they become activated, they need to it's a bit like a kid in the sweet shop. They need to get energy really, really quickly. And when they get that energy really, really quickly, they become bad. So that's how the immune cells work with regard to metabolism. So they change the way they use energy to help them remain activated and be bad in this environment. So the third aim was really to figure out, so these monocytes, is the metabolism bad, and can we reverse that effect?
How was the study conducted and what was involved? So
as Ursula has previously mentioned, in this study we collected blood from healthy control donors and also ORA patients. So all of the ORA patients involved in this study were recruited from the rheumatology departments at St. Vincent's University Hospital under fully informed consent, and these patients had active established ORA in accordance with the ACR classification criteria. And again, as Ursula mentioned, we collected these 2 cohorts, but also we collected another 3rd cohort of this unique individuals at risk who develop aches and pains, but to date have no clinical signs or symptoms of ORA. So once this blood was collected, peripheral blood mononuclear cells, or PBMCs, were isolated. These were then magnetically labelled and sorted to yield a pure population of CD14 positive monocytes. So these monocytes would have been circulating in these individuals and patients' bloods hours previously. So once we isolated this pure population of monocytes, we then ran a number of experiments looking at them ex vivo, so unstimulated, or else under the influence of stimulation with LPS.
What were your findings?
So getting into, I suppose, the findings of our study. So firstly, I suppose, to elucidate the role of these peripheral blood monocytes in ORA, we firstly noted that there is a significant increase in the number of circulating monocytes in ORA compared to healthy control. And then we ran an inflammatory gene array, and really what we need to kind of focus on here are these red bars, which indicate those inflammatory markers which were hyper responsive in our ORA monocytes compared to healthy control. And as you can see here, that's quite a lot. And just to, I suppose, pick out a few of these genes, we further validated 3 of them here, and you can see that these are significantly increased in our ORA-activated monocytes compared to healthy control. And actually what was interesting with the genes that we found was that this inflammatory signature was very reminiscent of what an inflammatory M1 macrophage would produce in the joint. So those cells that the monocytes can become when they get into the joint, you know, the dendritic cell, the monocyte, the osteoclast. So we think that these cells are perhaps primed or poised to become inflammatory macrophages once they leave the blood and enter the joint. So again, as Ursula kind of mentioned earlier about the kid in the sweet shop type thing. So these cells, we know that a kind of a greater inflammatory burden, which we've just shown that these cells are hyper-inflammatory, is often associated with a greater burden or greater demand for energy or ATP. So firstly, looking here at the kind of mitochondria. So we know that the mitochondria are the powerhouse of the cell. And really here, what we can note is that we get a mixture of these regular and irregular shaped mitochondria in our ORA monocytes compared to healthy control. But then just to, I suppose, further elucidate the role or the metabolic competence of these cells, we use this machine, which is called a seahorse flux analyzer. So the seahorse machine, all it does is it measures the 2 major bioenergetic pathways of the cell in real time. So you've got glycolysis and oxidative phosphorylation. And you kind of get graphs out like looking like this, where you can get a reading of how much the cell depends on one pathway versus another, or do they depend on both. So what we found here was that these cells have, you know, a striking increase in their glycolytic capacity. So in this metabolic pathway called glycolysis, which is known to be associated with inflammatory function of cells. So glycolysis is this pathway whereby it's quick, you get a lot of ATP. Immune cells love it because when they want to get, you know, activated quickly, they don't want to go through the whole mitochondrial respiration, oxidative phosphorylation. It takes too long. But the problem with glycolysis is it's quick, but they kind of burn out quickly. So they don't get a huge amount of energy at the end in comparison to mitochondrial respiration. But really here, what we're seeing is this boost in glycolytic capacity of our cells shown by these traces. And again, this is further paralleled by significant increases in some key glycolytic enzymes, such as PFKFP3 and one of the master regulators of cellular bioenergetics, which is HIF1 alpha. And just looking here then, I suppose, at the metabolic phenotype profile, you can really see this switch from an aerobic quiescent phenotype towards this hyper-energetic, hyper-metabolic phenotype in our ORA-activated monocytes. So we do see that both pathways are boosted. However, when we look at our ECAR to OCR ratio, we can see that it's actually in favor of glycolysis. So even though both pathways are up in these cells appear to depend more on glycolytic mechanisms than oxidative phosphorylation. And actually, when we inhibit glycolysis using 2DG, we see a complete abrogation of our inflammatory signature with this inhibition of this metabolic pathway. And not shown here, but when we inhibit the other metabolic pathway, we don't see this decrease or resolution of inflammation. So we were also interested in, you know, other regulators of this phenomenon. So STAT3 we know is implicated as a transcription factor. It's implicated in the invasive mechanisms of ORA. There's also been links between STAT3 and metabolism. So we thought this would tie in nicely. And when we looked for it, luckily STAT3 was significantly significantly enriched in our ORA monocytes compared to healthy control. And actually, when we block STAT3 using static, a small molecule inhibitor of STAT3, again, we see this resolution of inflammation. So we get a complete decrease of our inflammatory signature in the presence of this inhibitor. And actually, again, we get a decrease in the metabolic capacity of these cells. So this hypermetabolic boost that these cells were under, we can kind of return that to baseline in the presence of a STAT3 inhibitor.
We're discussing your recent study looking at CD14 positive monocytes in rheumatoid arthritis. What do your findings mean for individuals at risk, the people who don't actually have any clinical signs of rheumatoid arthritis at the moment?
So I suppose at this point, you know, we'd shown that these circulating RA monocytes were poised to produce these pro-inflammatory mediators. They had this hypermetabolic phenotype with what seems to be a reliance on glycolytic mechanisms, and also that this may be potentially regulated by STAT3. But we next wondered, you know, how early in disease does this occur? And so this is where we turned our sights to our individuals at risk, or our IAR cohort. So again, these individuals to date would have no clinical signs or symptoms of RA, but do have an increased risk of developing the disease. And really what was so striking here was that we're seeing that this hypermetabolic phenotype actually precedes clinical manifestations. And we see this stepwise increase from healthy control to individuals at risk to RA in our inflammatory panel. But actually what was even, so again, these monocytes are hyper-inflammatory. But again, what was really interesting as well is that the metabolic capacity of these cells were extremely similar to RA. And again, this precedes clinical manifestations. So we get nearly indistinguishable metabolic profile between our individuals at risk and our RA monocytes. And also we get quite similar levels of key glycolytic enzymes. And so the final kind of investigation in our study was, we were wondering what happened at the site of inflammation, so the synovial tissue. And again, we can see here this enrichment of this monocytic pathway genes in our individuals at risk and in RA in comparison to the healthy synovium.
How do you interpret your findings?
I suppose really kind of what we've shown here is that these circulating monocytes are primed for inflammation, they're primed for hypermetabolic mechanisms, and that this phenomenon appears to precede clinical manifestations. And to our knowledge, this is the first study to date to have investigated the role of peripheral blood monocytes in these individuals at risk. And I suppose studying these individuals may provide important clues or important insight into the evolution of the RA as a disease. I mean, as we kind of had up earlier, we've got this stratification of patients across the evolution of their disease. And really, if we can study these patients early, perhaps we can identify those who may go on to develop an RA and those who may not. And also maybe those who will respond to treatment and those who may not, which I know Ursula will elaborate on in a minute. But kind of the last thing I'll say about this is, this study also fits into the whole knowledge that perhaps monocytes are capable of building immunological memory, and this may be via mechanisms such as epigenetic imprinting, which is interesting in the context of RA, given the huge role that an environment plays in this disease, as well as genetic component. So this is something that we're looking to develop further in further research outputs for our group.
And pulling back and looking at this overall, what are the wider implications of your findings?
So I was going to finish off on that. So basically, we started off this podcast talking about those patients there with chronic rheumatoid arthritis under number 3. And basically, when we were treating patients at that stage, you're talking within 5 years, patients had, functional disability, and within 10 years, the majority of rheumatoid patients had given up work because of this functional disability. So we've come a long way over the last 20 years, and we've got some great targets now that target individual proteins or target individual cells. But still, I kind of divide this into 3 groups. With rheumatoid arthritis patients at the moment, what we have is a third of patients respond great to treatment. A third of patients have suboptimal responses with side effects, and a third of patients don't respond at all. Now, if we treat them early enough, we get better percentage differentiation between all those 3. But clearly, we have suboptimal responses in some patients, and we have no patients responding at all. And the other aspect of what's happening at the moment is that while we have all these great therapies, what happens with patients is they go into a clinic, they get a therapy, they might fail that therapy, then they get the second therapy, they might fail that therapy. And by the time they get to the third or fourth therapy, they have developed destruction in their joints, and it's irreversible. So what we want to do is to be able to give patients the right treatment on the day they arrive into the clinic at the very beginning, because we know the earlier we treat patients, the less chance they have of getting irreversible damage. So this, I suppose, study is allowing us, so the signature, we know the macrophage, which sits in the synovium, just as context, it's the only cell type that has shown as a biomarker in the tissue response to treatment, but we can't biopsy patients in a clinic. It's just people don't do it. So what this study has shown us that the signature that we're finding in the blood, which we could do in a clinic, seems to be reflective of those macrophages in the tissue. So what we're hoping is that if we expand this study into larger cohorts of rheumatoid patients, that it might help us to figure out what patients will respond to treatment or not, and a particular treatment. The second is that we're going to be monitoring the signature in those patients at risk, and that the minute they start to show signs of rheumatoid arthritis, that we can treat them early and again stop that irreversible damage. Some studies are actually treating patients even before they get, well, individuals before they get rheumatoid arthritis. And I suppose the final implication of the study is that for that third of patients that don't respond to any current therapies at the moment, we're suggesting possibly that this metabolic switch in the cells, if we target that metabolic switch, can we turn off the monocyte and then turn off the inflammation? So they're the three aspects I think that are really important. But the key message is that we want to find new therapeutic targets for patients that don't respond, and the earlier we can treat patients, the better their responses are, and we might achieve remission in more patients.